All right, welcome back. Today we're going to be starting our unit on genetics by talking a little bit about some vocabulary, Gregor Mendel, and then some monohybrid crosses. So first we're going to review a little bit about what we talked about our last unit, which was about types of cell division. So the first type of cell division we talked about was mitosis. And mitosis was involved in asexual reproduction. Now, asexual reproduction, again, only requires one parent. You don't need a partner. And it involves in making identical copies of a cell, which are clones of the original cell. Some of the pros of asexual reproduction are organisms look the same. You don't have to waste time finding a mate. It's often very, very fast and efficient. But the biggest con is that there's no genetic diversity, and genetic diversity is very important in the world around us. Genetic diversity, it actually helps species that are diverse survive for long periods of time. And the second type of reproduction we talked about was sexual reproduction. And sexual reproduction requires two parents. And the process that we talked about involved in sexual reproduction was called meiosis. And meiosis made haploid gametes. Remember, haploid means half the normal genetic information. Gametes are sperm and eggs. Eggs, again, have 23 chromosomes. Sperm has 23 chromosomes. When you add them all up, you get a diploid zygote. And those chromosomes, 46 in total, are going to be arranged in homologous pairs. And each of the chromosomes in a homologous pair, one comes from mom and the other one comes from dad. So you have one copy of each chromosome from mom and one copy of each chromosome from dad. That gives you two copies of each, making an organism diploid. Now, what we're talking about in genetics is essentially which genes you're getting from your parents, what combinations of genes are you getting, and how do those lead to the traits that you have. Now, that's going to take us into a man named Gregor Mendel. Gregor Mendel is known as the father of genetics. And he documented patterns of inheritance in garden peas. So he did a lot of work studying garden peas and how they inherited their traits. Now, you may ask, why the heck did he study peas? Like, why did he choose peas of everything? And the reasons that he chose peas were because peas, they're easy to see, and it's easy to study their traits. It's pretty much black and white type of traits. Is the plant tall or short? What color are the peas? What color are the pods? What color are the flowers? And typically it's an either or. It's either green or yellow. It's wrinkled or round. It's tall or short. So they were easy to see, but also, they had many different traits that you could analyze together. Examples include flower color, seed color, and seed shape. And again, that's not all of them. He also had height of the plant. He also had strict control over which plants mated with other plants. So he controlled the mating process. So he knew the traits of the parents, and he knew the traits of the offspring, and he controlled which ones mated with each other. Pea plants also have many generations in a short period of time. And there was lots of offspring per each cross, which gave him lots of data rather quickly to analyze. Now, some of the work that he did included cross-pollinating true breeding parents. Now, true breeding parents, it's kind of like it when we're talking about a purebred animal. So he would look at the parents, and he knew that parent 1 was pure white flowers and parent 2 was pure purple flowers. So he crossed true breeding, which is essentially purebred, plants with one another. He raised them and observed their traits in the offspring. After that, so he let a pure white flower and a pure purple flower, he mated them together. He observed the traits in the offspring. He allowed them to grow up. And then he self-pollinated them, meaning he crossed a purple flower from the first generation with another flower from the first generation. So he crossed these, self-pollinated them, and then observed the next generation, which would be the F2 or the second generation. So if I was to write that out, for P generation, he had pure white crossed with pure purple. He mated the white with the purple, and he observed the F1 generation. F1 again means just first generation, and all of the offspring were purple. Then he self-pollinated them, meaning he let a purple cross with a purple. He self-pollinated them, and he looked at the offspring, and this is where he found something interesting. He noticed that the F2 generation, 75% were purple, and 25% were white. And this was curious to him because when he crossed the purple flowers, there was no white parent. It was a purple with a purple. But he noticed that the white color came back. And it was very interesting because it skipped a generation. And he tried to figure out how exactly did that trait skip a generation. And this led to a lot of the work that he did, was this interesting pattern of a trait skipping a generation, and he was trying to figure that out. So this is what he came up with. For each characteristic, an organism inherits 2 different alleles. Alleles are versions of a trait. So he realized that there are traits come in different versions. So he knew that one trait was not simply one color, but there were different versions of that trait. Tall or short, green or, it wasn't green. It was yellow and green, or white, purple, or round or wrinkled. They came in alternate versions, and those alternate versions are called alleles. So alleles are the different versions of a gene, meaning it either had the allele for being a white flower or for being a purple flower. He also noticed that some traits masked others. So if you had one version of a trait, it could cover up another version. So if mom gave you one and dad gave you one and they were different, one could cover up the other. So in the example of purple flowers and white flowers, purple and white flower colors are separate traits and they do not blend, meaning if you get an allele for purple and an allele for white, they don't mix. But what does happen is that the purple color, purple, hides the white. So if you get one allele for purple and one allele for white, and you have both, the purple covers the white and the flowers are going to be purple. And what he called these were the dominant allele, the dominant allele is the one that is expressed, the one that shows up. It's the one that can cover up others, and therefore it's represented using a capital letter. The other allele, the one that is covered up, he called recessive. So the recessive allele is the trait that is not expressed if the dominant form is present, and it uses a lowercase letter. So essentially what he says is because purple covers up white, big A would be purple, little a would stand for white. You have 2 copies of each gene, meaning you have 2 letters. One letter you get from mom, one letter you get from dad. So if mom gave you a big A and dad gave you a big A, that's easy. Your flowers are purple. But if mom gave you a big A and dad gave you a little a, something interesting happens. The big A covers the little a, therefore your flowers are purple. However, if mom gives you a little a and dad gives you a little a, your flowers are white because you do not have the big A to cover them up. So this is what Mendel found. The dominant allele, represented with a capital letter, will cover up the little letter if it is present. It's almost like it's not even there. And this brings us into a few more vocabulary words, and those words are genotype versus phenotype. So the first word we're going to talk about is phenotype. The phenotype is the physical appearance. So essentially it is a description of the organism's trait, what it looks like. Your phenotype is your physical appearance. Tall, short, white flowers, purple flowers, round seeds or wrinkled seeds. It's what you look like. Whereas your genotype is a description of your genes. So it's your genetic makeup, the genes that you have. So the phenotypes for color is going to be, are your flowers purple or are your flowers white? The genotype for color are going to be, did you get 2 big A's? Did you get a big A and a little a, or did you get 2 little A's? So these are the genes that you inherited here. And the phenotype is what you look like. And we're going to be referring to both as we work. All right. So again, alleles can be represented as letters. The dominant allele is going to be the capital letter. The recessive allele is going to be the lowercase letter. Piece of advice that I give you is when you are working with a letter, I usually try to choose a letter that is different, capital and lowercase, like an A or a B, because they look different, capital and lowercase. If I'm using a letter such as a P, I make absolutely certain that I can tell if it's a big P or a little p, because I make my big P really big and my little p really little. That way you don't see them and you get confused. You don't want to accidentally confuse yourself. So dominant is capital, recessive is lowercase. True breeding for purple would be 2 big P's, and true breeding for white would be 2 little p's. What Mendel did was he crossed a big P with a little p, a purple, pure purple with a pure white. He got all the offspring and they were all purple. He allowed those to self-pollinate, and observed the next generation, which would be the F2s. So again, the P generation was purple mixed with a white. That would be a purple would be big P, big P. The white would be little p, little p. When he mixed those together and let those two mate, F1 generation was all purple. But their genotype was big P, little p. The reason he knew that is because this parent had to give a big P, this parent had to give a little p. So all the offspring had to be big P, little p. Then he mated the big P, little p's together and looked at the offspring. So this is going to lead us into how we make a Punnett square. Now, a Punnett square is a square that we are going to use to help us figure out the chance that the offspring is going to have certain traits. So what's the chance the offspring are going to be white versus purple? How many roughly are going to be white? How many are roughly going to be purple based on the genes that the parent has? So the F1 generation, again, we are self-pollinating the purple flowers, which were big P, little p. So both parents, parent 1 is big P, little p, parent 2 is big P, little p. When you're filling out a Punnett square, the first step, you got to figure out your parents before you can do anything. Now, we knew here that we are crossing the F1 generation, which was big P, little p. Now, we're going to fill out our square. Typically, the father goes along the top and the mother goes along the side. That does not matter. So when you're filling these out, if you're like, oh no, I accidentally put the mother on the top, it doesn't matter. As long as you fill it out correctly, it doesn't matter which side mom goes on or which side dad goes on, and sometimes you don't even know which one's mom and which one's dad. The way that you do this, you take parent 1, so let's say parent 1 here, parent 1, parent 2. We take parent 1, we'll say that parent 1 is dad, and we'll put his letters along the top, and we separate them because dad is only going to give one of these letters to his offspring. He could either give the offspring a big P or he could give them a little p. And we do the same for mom. Mom, parent 2, could either give the offspring a big P or a little p. And this chart is going to help us figure out what happens if dad gives a big P and mom gives a big P. Then it would be big P, big P. For this one, what if dad gave a little p and mom gave a big P? This is kind of like a multiplication chart. So this letter goes here, this letter goes here, this one here, this one here. Same down here, this letter goes here, this letter here, just like a multiplication chart. So just like a multiplication chart, and you fill in your letters. And this will let us know that this flower, big P, remember, big P stands for purple, little p stands for white. Our key, big P, equals purple. Big P, little p, remember, big P covers the little p, so this is purple. And little p is white. So we know that big P is purple. Big P, little p is also purple. Big P, little p, purple. Little p is white. And after this, we're going to look at the chance that the offspring are going to have what genotype or what phenotype. So looking at the genotypes of the offspring, that's going to be the letters. How many of our offspring in the square are big P? Well, there's just this one right here is big P. How many are little p? Well, we have these two are big P, little p. And then, how many are little p? We only have the one that's little p. Now, the phenotype is what they look like. How many of these are purple? We have 1 purple, 2, 3 purples, and only 1 white. And that is how we fill out a Punnett square. Now, another word that we're going to come across is homozygous and heterozygous. Homozygous. Homo means same. Zygous means alleles. So homozygous would be 2 of the same allele, which would be big P or little p. Those are the only options for having 2 of the same.